Hey class, this is Mrs. Wendt, and this is a recording of the notes that we did in class on Wednesday, January 5th. And this is a continuation from notes that we took on Monday. So we did part 1 on Monday. This is part 2. And in this part 2, we're going to talk about mainly this word translation, which is where we take the messenger RNA we made on Monday, and we're going to use that to make a protein. Okay, so just a quick recap on Monday here. We talked about making some RNA, which is here on the right, and we kind of went over the differences between DNA and RNA. And there's 3 kinds of RNA right here. So we had the ribosomal RNA that we're going to use today, the transfer RNA, which kind of looks like the letter T, and it has an amino acid on the top of it, and then the messenger RNA, which stays as a long line so we can read each letter. And the messenger RNA is made like this picture here that we discussed on Monday, where we have the 2-sided DNA gets opened up by this enzyme here called RNA polymerase. That enzyme also is going to match bases up to one side of the DNA, and that makes this blue messenger RNA. And then once that's made, it's going to leave the nucleus and go to a ribosome. So that's where we're kind of starting with today. Now at the bottom of the page, the last thing we did was we matched up letters. So we made this piece of messenger RNA. And this piece of messenger RNA is where we're going to start today. So I actually copied this and put it kind of at the top of this next page here. Okay. Now I'm going to click this link here as well. All right, let's kind of review all the steps with this animation here, guys. So we have our cell, and we zoom into the nucleus, and then here's our chromosome with our DNA, and we're going to take this one piece of DNA, so not the whole thing, but just this little piece here that we need a copy of, and we're going to make a piece of messenger RNA to copy that information.
These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of 3 bases in the nucleus. The DNA code determines the order in which the 3 bases are added to the messenger RNA.
Okay, so remember that the DNA had the letters C, G, A, and T. But for our RNA we're building, instead of the letter T, we're going to use the letter U to match up to the A's. So this is going to have C's, G's, A's, and U's. And we're only copying one side. We don't copy both sides. So this is happening in the nucleus because DNA has to stay in the nucleus. Once we make our copy, it's going to go to a ribosome.
This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid.
So you can see from the video here that at the bottom, this big long line of letters, this is our messenger RNA that we made in the nucleus that came out to this brown ribosome. And in the ribosome, the transfer RNAs will line up so the letters match up with the message. And each group of 3 letters represents a specific amino acid. So that way these purple amino acids get put in the right order for building. Okay, so like if you have a bunch of pieces of wood, brick, nails, and windows, depending on what order you put those things in, you could build a house or a garage or a tree house or, you know, a small house or a big house or whatever. So the order that these building blocks go in is going to determine what we're building. Okay, so we're matching letters up 3 at a time, and the groups of 3 letters are called codons. We'll have that vocab word in a minute. Remember, these are transfer RNAs. They kind of look like little letter T's in this animation. And these purple amino acids that we're putting together, when there's a whole bunch of them together, that makes our protein.
This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Now this folding part is really important too, because the way the amino acids...
5 seconds, you can skip this step.
Sorry about that. The way those amino acids are put together, the folding is going to make a big difference on the shape of the protein. And proteins can do their job by their different shapes. Okay, so let's go back to your packet here, guys. Here's the messenger RNA that we drew yesterday, or Monday, I should say. And the first thing we have to do is make what are called codons. So to do that, we are going to put these guys in little groups of 3. So these first 3 bases here, UCC, that is our first codon on our messenger RNA. So right down here, I would write UCC. And my handwriting is going to be horrible for this. I apologize. Okay. Then, without skipping anything, just right next door to this, here's my next group of 3 letters. So this codon would be G, G, U. Okay. 3 more letters, GAG, GAG, and so on. So your job is to finish the last 3 codons there. Okay, and after we break this all up into codons, sometimes you will have extra letters at the end, and you'll notice that in the lab. But altogether, there's 1, 2, 3, 4, 5, 6 different codons, and every codon will always have 3 bases, 3 nucleotides. Okay, so you need to finish these 3 here, and I know you can do that. So that's how we are going to match up these amino acids. Okay, so I can see here if this transfer RNA, if there's an AAA on the messenger RNA, I know up here that's going to match up with a U, because A's and U's match up. Okay, the next one says GAU. I know this has got to be a C to match with the G. This will be a U. This will be an A. Okay, so the letters are going to match up just like what we've been doing. Okay, now on this transfer RNA, AAA is the code for this specific amino acid called lysine. GAU is the code for this amino acid called aspartame. Okay, now we don't have to memorize these. There's always going to be a chart. We can look them up. But your next question on your paper, well, I think I'm getting ahead of myself, so hold on one second. Yeah, getting ahead of myself. But that's what this picture's all about. So let's look at your next questions here, guys. Under the picture, question 19 says, above is an image of translation on what organelle is taking place. Well, translation happens in the ribosome, and ribosomes are in the cytoplasm. Whoops, sorry about that. Okay, question 20. the amino acids get transported by the transfer RNAs. So they look like T's in our video clip. They don't really look like T's on the worksheet, but it's the same thing. Okay. 21. After the transfer RNAs drop off their amino acids, they're considered empty. So the transfer RNAs are kind of like little taxis, and we had these during photosynthesis and respiration too, where they pick up a molecule, take it to where it belongs, drop it off, and then their job is to actually just go pick up another one and do it again. Okay, so after the transfer RNA molecules drop off the amino acid, they're just going to go find another amino acid and repeat it over and over and over again. That's what they do all day long. Okay, so again, we saw that with photosynthesis. There's a lot of processes in your body that does this, where one chemical will just go pick up the missing pieces from the different body parts and bring them to where they need to belong. Okay, now in 22 here, it says, look at the messenger RNA strand above and write the codons. Okay, so the first one here, UGG, I copied it there. Go ahead and write the other codons in these spaces. And when you're done, the right answer is actually part of question 23. So finish 22 and then use question 23 to check your work. All right, so whoops, sorry about that, guys. Same picture here. There's our codons busted apart. And there we have something different written down. I wrote down the symbols for the amino acids. So I looked on the transfer RNA and I saw, okay, UGG, that must be the code for this guy right here. AAA, that's the code for lysine. GAU is the code for this guy, aspirin. Okay, and the amino acids all have a 3-letter abbreviation to them, kind of like all the elements on the periodic table. So biologists will always use these abbreviations. You don't have to memorize them, you can always look them up. Okay, but so I just matched the 3 letters from the message to which amino acid there was, and that's what I wrote on these blanks here. So that's these 4 amino acids matched up with those 4 pieces of the code. Okay. Question 24. What's the responsibilities of transfer RNA during translation? Okay. There's lots of ways to write an answer for 24. Here's something that we came up with during one block yesterday. We said transfer RNAs have to pick up an amino acid, bring it over to the ribosome, and then the transfer RNA has to match the base letters to be sure to be in the right order. So there's a lot of different ways you can answer 24. There's just one example right there. And if I ever go too fast, remember, you can always pause the video to write if I'm going too fast. This big circle here, we're going to get to that right now. Okay? So what this circle is anytime you have a set of letters, okay, so let's say I have AUG as a set of letters on my RNA code. I can look on this wheel and see which amino acid AUG is always going to mean. And this works for humans, but also all other living creatures, because we all use the same DNA code, which is pretty amazing that all living things on Earth use the same code there. Okay, so to read this, the first letter of the code, I'm going to start in the center of this circle here. So my first letter is A. So I'm going to go to the center of the circle and I'm going to find A. what are you doing here? Hold on one second, guys. So here's A. The second letter is U, so that goes in the next row. And then the next row, I'd go to my third letter, which is G. And that shows me that MET, or MET, is the amino acid that AUG codes for. Okay, so let's try another random one. Let's do GGG. Okay, so if I have GGG, I'd start in the center here with the first G. I go to the next G, go to the next G, and that is going to code for glycine. Now, if you notice that GGA also is for glycine, GGC also for glycine. Okay, so there's actually 4 different codes that are going to give us glycine. And that's true for a lot of amino acids. There's 20 different amino acids, but there's a lot more than 20 letter combinations. So many letter combinations mean the same amino acid. Okay, let's do one more example here real quick. Let's say we have UGA. So here's a U, here's the G, and here's an A. Now this one's a little weird. There's a black dot there. Okay, so what that means there, UGA, doesn't actually code for amino acid. Instead, it's a codon that means stop. And that tells the ribosome to stop building and release that protein out into the cytoplasm. And there's actually 3 of them. Okay, UAG and UAA also have those black dots that mean stop building. Okay, so you're going to do some practice with this wheel. And there's actually another type of chart that's a square. And when you come back to class, I'll show you how to use the square chart. It's the same info. It's just some people prefer the circle, some people prefer the square. All right, so now on your worksheet, it says use diagram 5, so that's the circle. We can determine what the amino acids are being coded for. Use the chart to find out what the code AUG codes for. Now that's one we actually already did. We started with that. AUG codes for the amino acid MET. So in this blank here, I would just write MET for MET. Okay. So you're going to be kind of using that chart to figure out all of these guys, and then you're also going to write down the 3 that means stop. Okay, so we figured out one of them that means stop was UGA, because that had that little black box. It's also going to be UAA and UAG. Okay, so one of them is going to be UGA. Right, my dyslexia is making me, yeah, UGA. And then you can go ahead and fill in the other 2, that means stop. And then you're going to go to your circle and look up all of these guys. So we already did AUG, that's MET. UUC, find that one here. Let's see, UC would be that guy right there. So this one is PHE. So you always start from the center and work your way out. So go ahead and try the next 4 and see how it goes. So you can pause the video and try those real quick. All right, go ahead and check your answers there. That's what I got using the wheel. So if you made a mistake, see if you can find it. And again, if I go too fast, you can always pause the video at any time. All right, next page we have a diagram here. And this diagram kind of shows the difference the three kind of things we're going to model with the kit, we did replication before Christmas, transcription we did Tuesday, we're going to do translation on Thursday. So replication is when you start with a piece of DNA and you make a copy, so you end up with a copy of DNA in the end. And this circle goes around and around to kind of indicate that you're recycling, you're using the same piece of DNA over and over and over again. Okay. Transcription is a little different because you take the DNA and you're going to make an RNA with it. And that RNA is used just one time. And then during translation, that RNA gets changed into a protein. And again, the RNA only is used one time and then it's destroyed. Okay. So at the bottom here, it says replication. We're going to start with a piece of DNA. And at the end, we're going to produce, we're actually going to have 2 DNAs at the end. Okay. So you need to use that diagram above and fill in these other blanks. You're going to use words like protein and RNA and just kind of following the picture here. So make sure you fill out 28 before you go on. And then question 29, the DNA replication shows a circular arrow, kind of like the recycling symbol, but transcription and translation have a one-way arrow. So what I tell my students is the DNA is used over and over, but RNA is used only once. And that's a pretty good answer for number 29. You could absolutely put that in your own words if you want to, though, too. Number 30, add the location of each process to diagram 6. So if we go back to diagram 6 here, replication is copying the DNA. DNA can only be in the nucleus, so that has to be in the nucleus. Transcription also has DNA involved. DNA never leaves the nucleus, so that has to be in the nucleus. But translation is going to happen at a ribosome. Okay. So we did those locations. So I can check that off because we put those on our picture. Then, why do all these processes in diagram 6 happen? What's the goal? The goal is to build proteins. So we need proteins built for all of our body parts and many chemicals our body uses. Okay, so proteins are very, very important for everything that we do. Your body is always building more proteins because you always need more cells to repair things and to replace worn out cells. You need to fix injured body parts, but you also use up a lot of chemicals to digest your food, hormones that make your body change in different ways, and all of those are going to be proteins also. All right, so this back page, I'm going to get you started, but then you're going to finish this and then hand it in. So when we're doing DNA replication, we know we're going to match letters, but you have to remember that if we're doing DNA, the letters we're going to use for DNA are A's, letter T, C's, and G's. Again, bad handwriting, that's okay. But down here for RNA, we're not going to use the letter T. We're going to use letters A, U, C, and G. Okay, RNA uses the letter U instead of G. Okay, so if I look here for the DNA, T is going to match up with an A is going to match up with a T, G will match up with a C, this C will match up with a G, G with C, C with G, and so on and so on. You can finish that. Okay. For RNA, it's going to look really similar. T's going to match up with A, but right here, this A, we're not going to build a T, we're going to build a U. The G will still be a C, C will still be a G, and so on and so on. So you're going to finish building those guys. Then we're going to take our RNA that we're building. We have to bust it into codons. So the first 3 letters here, A, U, C. That's a very bad U, I apologize. Okay, so that's our first codon, AUC. So you write the codons for all the rest. And then I need to look on my wheel here, AUC. AUC. That is going to be this guy here, lysine. Not lysine. I can't remember the name, but it's I-L-E. So capital I, lowercase l. So that looks kind of weird, but so I-L-C. That's the amino acid that codes for. So you'll write down all the 3-letter combinations that you wrote up here and then match them up on the wheel. Okay? And that's also what you're going to be doing in lab on Thursday. So that's it for these notes here. So what you need to do is fill in the whole notes packet, ask if you have questions. So you can come into my advisory, my after school, send me a message, send me a video, check with a friend, whatever. Okay? Because you are going to hand it in. All right? Now, with this notes packet, there are 2 labs, and they are both quiz grades. So it's very important you do both labs. Okay, so I'm going to show you, for those of you that are absent in Schoology, kind of what to do about those lab issues, okay? So in our Schoology folder, Unit 5 here, there is the lab. Bam. So here's the lab, the first one from Tuesday, and then here's the one for today. And you can put the file for the lab questions in Notability, but there's this thing here called Video Playlist. And if you open the video playlist, there's 5 videos. And the first 3 videos go with lab number 1. So you can see it's DNA transcription 1, transcription 2, transcription 3. So those we did on Tuesday, and then Thursday we're doing translation 1 and translation 2. You can watch the videos. That's me and Miss Mills doing the lab, and you can fill in the lab questions watching the videos. Okay, I know some of you guys are out sick, but you've got to watch the videos and fill in the lab questions because there's no test for this unit. The labs are your quiz for the unit. Okay, so again, please let me know if you have any questions, and you know, don't be a stranger. Send some messages and stuff. The end of the quarter is close. I know it sucks if you're out sick, but just get it done as best you can. Have a great day, guys.